Hello everyone, today we'll be learning about a novel method of drug delivery involving red blood cells as extracellular vesicles. Now, to start off with, what are red blood cell derived extracellular vesicles? Well, these vesicles are a type of extracellular vesicles which are cell-derived membrane-surrounded vesicles that carry bioactive molecules and deliver them to recipient cells. As you can see from the diagram, the types of bioactive molecules that they carry can range from proteins and nucleic acids to even lipids. Classic kinds of EVs include exosomes, microvesicles, and apoptotic bodies. The most commonly known type of EV, exosomes, is formed through the following mechanism. As you can see, exosomes are formed via an endocytic pathway through the invagination of endosomal membranes, which form multivesicular bodies, also known as MVBs. These MVBs can fuse with the plasma membrane in order to release exosomes into the extracellular environment. This process results in the creation of two types of EVs, exosomes and microvesicles. Exosomes, are shown in the diagram, are formed within the cytosol of the cells, within the multivesicular bodies, whereas microvesicles are formed near the plasma membrane through the outward budding and fission of the plasma membrane. Now, EVs are typically used in the research to study their ability to act as carriers of biomarkers for diseases. Biomarkers are biological molecules found in the blood or even other bodily fluids or tissues that indicate a normal or abnormal process, condition, or disease. A biomarker may also be used to monitor how well the body responds to a certain treatment. The fact that EVs can deliver biomarkers mean that they are oftentimes used in disease detection and hold great potential in that specific industry. Now, to give you an example of their application, exosomes isolated from the urine have demonstrated their ability to reflect acute kidney injuries. These exosomes hold great potential for the diagnostic industry because they're found within bodily fluids such as blood and urine. This means that disease can be diagnosed and even monitored through minimally invasive liquid biopsies, which completely eliminates the need for invasive surgical biopsy methods. But beyond serving as biomarkers, EVs hold great promise for becoming the next generation of theragnostic nanoplatforms for drug delivery. The reason is that EVs are highly biocompatible and relatively less toxic due to their natural origin. Thus, they're oftentimes preferred for drug delivery purposes over synthetic vehicles and nanoparticles. On top of that, there is clear evidence that these EVs can cross multiple biological barriers. For instance, EV cargo remains stable even in highly acidic environments of the stomach. Research published in the MDPI on extracellular vesicles carrying the miRNA 150 as a potential suppression method for delayed type hypersensitivity, underlying casein allergy revealed that delayed type hypersensitivity was successfully reduced by orally supplemented miRNA 150 IBs. Now, this diagram summarizes the different biological barriers that can be penetrated by the EVs. And that brings us to the end of our video. Thank you for watching, and in our next video we'll discuss more about red blood cell extracellular vesicles.